Good evening, everyone. My name is John Measy, and I just want to start by saying that I'm very sorry that I'm not able to be with you live this evening as I had expected. Unfortunately, I've been called away on a very, um, according to my boss's, very important mission that I have to attend. So I've recorded this presentation for you the day before. I think you'll find that it'll be exactly the same as if I were here live. So as I said, my name is John Measy. I am a professor of invasion biology, and I have two separate jobs, both at Centers for Invasion Biology in Yunnan University in China, where I am now, and Stellenbosch University in South Africa. So in this presentation today, I'm going to give you the historical context for this invasive species, the African clawed frog, or its Latin name Xenopus laevis. I'm going to talk to you a little bit about the phylogeny. That's the sort of the family history of this species as we know it. And I'm going to then talk about present day invasions, where they are all over the world, including in Arizona, and some of the research that's been done showing the rapid evolution of these invasions, and in particular in the French population. Then I'm going to talk about some of my own work on the gut microbiome of Xenopus laevis. This work, I have preliminary results. It's ongoing. I thought I would share them with you. And then lastly, I'm going to give you a quick overview about what we know about the established population of Xenopus laevis in Arizona. And so our story starts in the 1930s in a remote corner of South Africa. This little red blob shows you the very southwestern tip, which is shown in this bigger map, and a very, very small valley where this red dot is, showing where Yonkers Hook Nature Reserve is. Now, at the time, this small valley was the center of the Nature Conservancy Council of the whole of the Cape region. And back then, this included more than half of South Africa as we know it today. And in this area, in this headquarters of the Nature Conservation, was the head of this was a man called Douglas Hay. And the 1930s was, I wasn't alive, I wasn't around, but I think it was a very optimistic time. People felt that you could change the world just by doing a few small things, and the introduction of species was seen as a very positive thing at the time, and Douglas Hay was responsible for introducing various alien species of fish all over southern Africa. So he raised fish in the fish hatchery in Yonkers Hook Nature Reserve. They have a beautiful clean water supply, and he constructed large concrete tanks where fish were raised, and then the offspring of these fish was sent to various different colonial areas in southern Africa. So as far north as Malawi, including parts of then Rhodesia, what is now Zimbabwe, and virtually all of the high altitude streams in southern Africa. And at the same time, in the University of Cape Town over here, researchers were looking at a frog that is very common in these areas called the African clawed frog, or Xenopus laevis, and they were using this species to do research on development and endocrinology, because they found that you could breed it all year round. But it wasn't until a British researcher came from London and spent some time working with these guys in South Africa that his name was Lancelot Hogben, and he took this information about the ability to breed the species back to London and published an article saying that it's very possible that you can now use this species to look for pregnancy in women, because it's possible to keep this frog in a laboratory year round and use it to look for the presence of luteinizing hormone in the urine of pregnant women. A little bit of background there. So this was quite well known in University of Cape Town, but they hadn't made the combination between the need for a pregnancy test, which is what Hogben was looking for, and the frog that they were using for their endocrinological experiments. So Hogben, to put these two things together, and so the pregnancy test using the African clawed frog was born in the late 1930s. And this created a lot of excitement. First of all, Hogben became very famous, but it also meant that there was a sudden desire to have colonies of these frogs in laboratories all over the world so that doctors could use them for pregnancy testing. So up until this point, there have been various attempts to use different animals for pregnancy testing, including rabbits, rats, and some European amphibians, including the common frog. But all of these models suffered from the fact that they had distinct breeding periods in spring, and then you couldn't detect the presence of luteinizing hormone in a pregnant woman at other times of the year. So the advantage of the African clawed frog was that at any point during the year, you could inject urine collected from a woman who thought she might be pregnant into the dorsal lymph sacs of this frog and put it together with a male. And if luteinizing hormone was present, then the female frog would allow the male to mate with her, and in the morning there would be a lot of eggs. And so the pregnancy test was really the presence or absence of eggs the following day. And technicians all over the world, frog technicians, became very familiar with this species, and it was used successfully to diagnose pregnancy from the very late 1930s all the way through to the 1970s, when other tests became available that didn't involve having to keep colonies of live frogs. And so the genesis of the movement of this species around the world was through this kind of pregnancy testing. And Douglas Hay felt that it was his duty as somebody who had the facilities to breed these animals to actually breed them and share them with the rest of the world. And so that's exactly what he did. He created pens for breeding these, and that's what we see some colleagues of mine sitting in here. They are about to collect Xenopus from a dam that's just over here. And what you can't see here is that under all these plants are the old concrete tanks that they used to rear Xenopus in. Unfortunately for Hay, there was such a massive demand for these animals that he couldn't breed them fast enough to supply the demand. And so what he did was start asking local farmers whether they could actually go and take animals from their dams. And we know quite a lot of information about this because at the time, this was British Southwest Africa, and that huge amount of administrative data was kept carefully logged. So every time a truck went out to collect frogs, they would make a note of how much diesel was used. And from that information, together with a colleague of mine called Lance van Stittart, we worked out that they were actually going from Jonkershoek over into these mountains up here to collect frogs from lots of dams all around the area. Radius of about 50-60 kilometers. And it did look among this data as if towards the end of the period that they were exporting these animals in the 1970s, that they were having to go further and further in order to meet the quotas that they were selling these animals. And the other point about this story is that they were actually selling these animals at cost. So they weren't making any profit on these. Douglas Hay felt that he was doing the world a favor by exporting these animals for research. And because of all of the meticulous note-taking, what Lance and I were able to do is put together lots of maps using this historical data to show exactly where the animals were exported to. So what you can see in this map here is the large dot on Cape Town, and this is because they didn't only export animals to other parts of the world, they also were providing animals to universities in Southern Africa. So these circles here, these brown circles, show animals that are moved to London, Edinburgh, and Boston in the 1930s. Okay, and these would be for research purposes. And the two green stars are because in this paper we were also interested to see the correlation between these movements and the appearance of chytrid fungus. This is the amphibian killing fungus that about 20 years ago, Xenopus laevis, the African clawed frog, was implicated. The spread of this species was implicated in the spread of the fungus. And what you can see, what you will see quite conclusively, is that there's no correlation between where these animals were moved to and the appearance of the fungus. So those are historical data on the appearance of the fungus in Central Africa here. So by the 1940s, what you can see is that we also have some other symbols here, and that's the brown squares. Now the squares indicate where papers were published at that time using this species. So what we can see is that even though Douglas Hay and his colleagues were only exporting a slightly expanded area in Europe and in Eastern United States, at this point, other people were starting to get incidents, they were starting to get colonies of these animals, presumably shared by the scientists in India, actually in Australia, although it's under the picture of me, and in the Western United States. So there were colonies of these animals starting to be moved around the world. The other thing we can see is a single red triangle. So in 1944, the first extraliminal population of Xenopus was found on Ascension Island. It's hard to know exactly the reason why it got there, but Ascension Island used to be a point in the mid-Atlantic where airplanes would stop and refuel before they flew on to the United States. And so what we presume is that some of these animals that were being exported to the United States, perhaps in the 1930s, perhaps the early 1940s, were dispatched by some of these people, perhaps because they had changed plans, onto Ascension Island. And we know that by 1944, there was a colony of frogs there that persisted for 20, 30 years. So in the 1950s, what we can see is the first examples of papers being produced in California using these animals, and also there's an export to San Francisco here. Meanwhile, a lot of the rest of the world, a lot of Europe certainly, and North America are also using these animals. And by the 1960s, we know that there was the first invasive population in California. Also, Arizona is appearing here for the first time, and we know that there was a release of animals in Florida. And this takes us to the 1970s, where you can see that the numbers of animals that are actually being shipped around the world are now enormous. So all of the United States seems to be receiving these animals, a lot of Europe, Japan over here, and also several examples in Australia. And the number of invasive populations is also popping up, creeping up, and including population here in Portugal, the population in France is implicated here, and populations in the UK that you can't see under these. Oh, you can see here, okay, in South Wales. So this was the end of the historical data that we could glean from the records of the Western Cape Nature Board. And shortly after the 1970s, the exports of Xenopus laevis stopped from that particular facility. They found that they just couldn't keep up with demand, and at the same time, two private companies in Cape Town had also started shipping animals, and they didn't keep records, or we haven't been able to get their records, and so after 1970s, we have very scant data about where animals were exported to, although we know that there were continued exports to the United States, Europe, and Japan after that time. And at that time, we're now moving on to the phylogeny of this species. At that time, in the 1970s, Xenopus laevis was the name for a huge group of what we now know are different species of Xenopus, but basically they thought that they occurred all the way from Cape Town up into a very southern part of Ethiopia here, and all the way over into West Africa. So at the time, they thought that all of this was Xenopus laevis, and that there were various different subspecies, and those you can see marked here in different colored polygons. And it wasn't really until the work of a colleague of mine, Ben Evans, and his PhD student at the time, Ben Furman, where they actually managed to collect samples from all of these different populations. And you can see the dots here, all the samples that they had. And then they sequenced a number of genes, both nuclear and mitochondrial, and put them into a phylogeny where what we can see now and recognize as different species are showing out to be completely distinct clades, okay, and quite deep divergence times. So these are the times here. This is 2.5, 5 million, 7.5, 10 million years ago. And so what we can see is that Xenopus laevis, which is this branch here, is distributed only from Malawi down into Cape Town, and within that distribution has a number of different clades. Okay, so the number of different families that apparently don't interbreed that much within this southern African area. And so what we now know is that Xenopus laevis only occurs within this kind of yellow polygon. And we know a little bit more information now. So up here, the distribution isn't continuous, but is confined to the highlands, and in the lower hot areas in these parts, these animals don't occur. Now, together with Ben and some students, we wanted to have a better picture of these different populations in southern Africa, And so we did some more work just on Xenopus laevis this time, and we made a collection, lots of collections of animals to see exactly where those clades occur spatially. And so what we can see in this figure is this is a different population called Poweri. Here is Peters' here is Borealis, and laevis is confined to this southern African area. And the different clades here are shown in different colors. And so the yellow clade, you can see, actually covers most of this area here, with a little bit of pollution, if you like, from the brown clade. We now know that these are actually all yellow clade animals, and it was just the particular gene that we were sequencing was similar to the brown clade animals. And then as you go into the mountains in southern Africa, what you find is that there's a lot of genetic differentiation. And so just moving over here, this is the southern end of this yellow clade, this clade that covers most of the distribution. But there's two lines that I'd like you to look at here. The dotted line here shows the end of summer rainfall, which covers all of this area. And this area we call, in Southern Africa, is known as the all year round rainfall zone. And it doesn't mean that it actually rains all year, it just means that it can rain at any time during the year. Okay? And then the solid line shows where the winter rainfall region starts. And so inside this solid line, we have only rain in winter. Inside this dotted line, we have rainfall in winter, can be in summer, can be at any time, and it will vary from year to year. And so if you're a frog and you're living in southern Africa and your populations are continuous throughout this area, then you need to be able to adapt and lay your eggs when the rains start. And so it's not very surprising that somewhere between these rainfall zones are where the frogs start changing in clades, because if you are breeding, if you're a summer breeder, then it's unlikely that you're going to mix with the animals that are winter breeders. And so we can see perhaps from the frog phylogeny where the true line of summer to winter rainfall occurs. The lines here are based on geological data from another publication. Okay, so inside this winter rainfall zone, we can see that we have several clades, and here is Jonkershoek. So here's the area that all the animals were exported from, and we can see that they definitely all were exported from what we call this blue clade. Okay, so populations all over the world all came from this blue clade, and we'll circle back to this information a little bit later in the talk. It's worth pointing out here that this little purple clade here turns out to be the most divergent clade, so it's the most different clade from all of these families, and we're estimating a divergence time of about 1.5 million years for this population here. And this population lives on top of a plateau. Okay, so there's a big cliff right at the front of these populations here of over 800 meters. And it seems that this clade was able to persist even when local other clades sort of disappeared and dried out. So you have to think of this area as undergoing multiple periods of drying and wet periods. And those would have related to expansion and contraction of these different populations. And we're talking about glacial and interglacial periods here. The last thing that I want to mention about these different groups is that the distribution of this species now on southern Africa includes pretty much all of this area. So Xenopus laevis has been found pretty much in all of this area, even though this is the Kalahari Desert. And so what happens is that because people have moved into this area and they've created farms, the first thing that they need to do is create a dam to sustain water. This is a very arid area. And this picture shows you just how arid it can be. So here's Cape Town down here. This is the Kalahari Desert, all of this, and this area is the Namib Desert. And these are very low rainfall areas. And so the distribution of this species goes up here and up into Malawi and then down. So you can see that actually this species is an arid adapted species. We tend to always read about Xenopus as being a Mediterranean species because this very small area of the Cape is a Mediterranean area. It has winter rainfall and hot summer dry periods, but the vast majority of the range of this species is arid. And so this species has lots of adaptations for living in hot, arid areas, and we'll circle back to some of those later. And so because people made lots of dams all over this area, Xenopus actually was able to move into a lot of these dams, and probably has a distribution much greater today than it would ever have naturally been before people came and started farming in these areas. So we've done quite a bit of research on this species in southern Africa, and I just wanted to touch on a couple of examples of that. This is a PhD student, Laurie Araspin, who worked both with me in Stellenbosch and my colleague Anthony Herrell in France. And with Laurie, what we did a few years ago was we went and sampled these animals along an altitudinal gradient. Okay, so not only is this species arid adapted, capable of living in really dry areas, but it occurs at 3,197 meters above sea level in Lesotho. Okay, Lesotho is a tiny little country in southern Africa. And so we basically sampled around sea level at 1,000 meters, at 2,000 meters, and over 3,000 meters. And we took these animals and we brought them back to the laboratory, and Laurie spent a very long time in temperature-controlled rooms chasing animals in circles in an experimental arena to find out how far they would move before they became exhausted. And that is what this data is that you're looking at now. And so she tested them at different temperatures. So here we can see temperature on the bottom. These bottom marks are actually not the performance data, but these are what we call CT max here and CT min. And to do these, we basically slowly heat frogs up in a chamber until they lose their writing response. So we don't kill them. This is not boiling frogs in water, but basically every couple of minutes and every degree that they go up, we flip them over, and if they stop being able to move back again, then this is a sublethal temperature at which they can't write anymore. And we can see that this is very consistent for the upper temperature bounds. So this happens just below 30 degrees, okay, for all of these populations, no matter where they come from. At the lower level, you can see that this is actually very different. And it corresponds directly to the altitude that they come from. So the animals at the highest altitude are able to withstand temperatures just above 4 degrees, whereas those from sea level can only withstand temperatures just under 6 degrees. So we can see that there's this variation of minimal temperatures that the animals can stand before they start losing their righting response. And at that point, we stop cooling or heating the water, and we gently bring the animals back to a normal temperature, a lab temperature, which is around 20 degrees. So these are all non-lethal experiments, let me stress that. So at a very cool room, at around 7 degrees, Laurie chased these animals from different populations. Each point here represents 20 animals that she's chasing around in tracks. And this is on this y-axis, we see the relative maximum distances that these animals were able to travel. Total distance is typical. Somewhere up here, they were traveling around 800 meters. So she's doing a lot of chasing around in circles. This represents this graph, this single graph in the paper here represents a huge amount of work that Laurie did. But what you can see is that animals from the sea level have this curve here that corresponds with a sort of right shift. So their optimal temperature is around 21 or 22 degrees Celsius, whereas animals from the highest altitude areas have an optimal temperature of 16 or nearly 17 degrees Celsius. And so what we can see is that within these populations, there is an adaptation to live either at altitude or down at sea level. And this is the temperature again in Celsius of this highest population here is 4.26, whereas the lowest population is nearly 6 degrees Celsius. We also managed to put temperature loggers into these ponds where we collected the animals from. And this is the pond at the highest elevation in Lesotho. And what we can see here is that for most of the year, all but about 4 months, for most of the year, this pond is below 10 degrees C. Okay, so it never goes to freezing because this probe is sitting at the bottom of the pond. Ice will form, thick ice will form on top of the pond and stop the water underneath from freezing. So the animals are able to escape freezing. But they really have a very, very little time during their year where they're above 10 degrees C. And so this sort of adaptation is totally necessary for them to be able to do all the things that they need to do when the temperatures are highest. Also note that when temperatures are highest, they also get quite low. So this is going down below 4 degrees C here, even during the summer when temperatures are really high. Meanwhile, at the lowest altitude, what we see is that temperatures virtually never dip below 10 degrees C. So there's a curve here, there's our 10-degree line, and at the lowest altitudes, the frogs are always in a habitat where they are comfortable and can move and do lots of different activities. At those low altitudes, this is the pond where we collected the animals. You can see that when we first arrived, there were a herd of zebra. There are some warthogs over here and lots of African wildlife around. We had to shoo a herd of water buffalo from one of the other ponds that we sampled, and giraffe and stuff moving around. So this kind of area is very exciting to work in. And we put traps into the water to collect the frogs overnight. This is the high altitude site in Lesotho, and you can see Laurie and another student standing over here. And so you can see it's very, very different kinds of habitats involved here. And so this is really quite extreme. There are very few frogs in the world that are able to have distribution, continuous distribution throughout this kind of altitudinal range. We also, with that other student that's here, Carla Wagner, we also did a study on the tadpoles, this time animals at 2,000 meters and animals at sea level. And what we found was very, very similar, but that tadpoles are actually able to withstand even lower temperatures. So this is 2.5 degrees Celsius, and this is 4 degrees Celsius for the sea level animals. So what we see is that all the life history stages of these animals have actively adapted to these different altitudinal ranges. And one of the things that tells us is that in its native range, this species has a phenomenal ability to adapt and change the environment that it's living in. Some data that I'm not able to show you here is that for her PhD, Laurie then went on and bred animals from these different populations, high and low altitude, and took them through to F2 generation. And what she found is that some of these adaptations actually remained in the F2 generation, so we presume that they're genetically fixed. But many of these adaptations actually disappeared when the animals were bred in laboratories over 2 generations. And that tells us that some of these things are actually plastic movements that the animals are able to do over time. So let's turn now to look at the present day invasive populations of Xenopus laevis that we're aware of. We know that habitat suitability models suggest that animals should be able to withstand all of this southern Australian and New Zealand areas, but it seems that animals haven't been introduced there, even though they hold animals in laboratories. There are multiple invasions in Japan, and I visited those this year. There's also 2 populations that we know of in China, but this animal is sold as a pet in almost every city in China, and it's expected that there are actually many more populations that we are unaware of there. So this is our origin, again in Yonkashuk, and we know this population on Sicily here at the end of Italy, population in Portugal near the capital Lisbon, the population in France I've already talked about, but in addition there are 2 more populations in France not shown here, one up near the border with Belgium that has actually crossed the border into Belgium and is currently expanding its range. There's another population down in Bordeaux where we hope the population is confined, but the landowner has now refused entry onto the land for anyone to go and check, and the French are now having to go through legal channels to access this. 3 populations shown in the UK here, all of which appear to now be extinct, although people are still following up with these. So this is the European situation, the Asian situation. The world's largest invasive population of this species is in Chile. This covers absolutely enormous area, tens of thousands of square kilometers. This started in Santiago, and there have been 2 dispersal events taking this up into a town called Copiapó, and I visited there about 1.5 years ago, and that's in the Atacama Desert. So these animals are living in areas for grape vineyards, where they actually grow wine in the Atacama Desert, and these animals seem to have moved into the dams there. We don't know how they got there. It's certainly transported by people, probably from this central invasion here. And of course, then we have the United States. Note here that Florida is missing. So the animals that we know were released in Florida don't seem to have started an invasive population there. There are a number of captures that have happened in Florida, but following up, nobody has found any invasive population here. And the one population near Tampa that I visited last summer turns out to be Xenopus tropicalis and not Xenopus laevis. So Xenopus tropicalis is from this area of West Africa. And that population in Florida is now expanding. It's using a lot of drainage areas to expand, and the population is growing in Tampa. But it's not Xenopus laevis, it's Xenopus tropicalis. So over here we see the population in Arizona, and here populations in California. The population that was in San Francisco appears to have been at Golden Gate Bridge Park. That appears to have gone, but populations in Los Angeles all the way through to San Diego are very much still there. I was there sampling last summer, and they're actively spreading. And so these have also moved across a national boundary into Tijuana in Mexico, and we sampled animals 60 kilometers south of the border in Mexico last year, and it's believed that they are still moving south into this Baja California area. And so I want to tell you a little bit more about the population in France, because this turns out to be quite an interesting story. So my story with this population started in the early 2000s when I was a postdoc in Paris. And I obviously had done a PhD at that point on invasive populations of Xenopus. And so when I heard that there was an undergraduate student who had found an invasive population, I immediately made contact with him, and we started working together on trying to get data for the paper that was eventually published in, I think, 2006. And so the story of this invasion, which is this black star represents a tiny little village in Western France, was that the French research authority, CNRS, decided that it would be a good idea to have a breeding facility to breed these animals. And I don't know exactly the reasons why it ended up in this small village, but a single gentleman was responsible for breeding these animals, and he built this facility over a small stream. So he had fresh water coming through, and he supplied the CNRS with animals for about 20-30 years. And so you can see that, and we know that in the United States there were also suppliers. I think they're based in Florida, but there are a number of suppliers around the world that bred animals and supply them to labs, as well as animals being exported from Southern Africa. And so this is a picture of that facility in this small village. And from the 1980s, locals who were fishing in this little pond, which has a stream directly up to this same house that you can see up here in the background, that's the same one, started angling. They were angling in this pond, and they started catching live Xenopus. And so it seems that this facility was never particularly secure. They had animals, either tadpoles or adults, jumping into the stream and coming down and colonizing this pond here, certainly from the 1980s. But from the early 2000s, Antoine started finding these animals in ponds outside this village. And so you can see on this map, any small dot is a pond where Antoine has sampled but didn't find animals, and any round circle is a positive pond. So a pond he put traps in and actually captured animals. And this is around 2002, this data. So you can see that in around 20 years, this species has managed to invade actually quite a considerable area of Western France here. And we can see that this is not a circular expansion, but the animals seem to be making their way up this river valley. by 2004, Antoine was only sampling outside of this same area that he'd sampled in 2002. And what you can see is that in the very small amount of time, this animal is marching, and it's now present in lots of ponds that it was previously absent from. And so we can see that in the early 2000s, there's a big expansion of this population. And the invasive range, at the time when we wrote this paper, it was published in, I think it finished in 2005, we published it in 2006. The invasive range was estimated at around 200 kilometers squared. And Antoine and I contacted a number of officials saying, look, this is clearly an ongoing invasion, it's a big problem, something needs to be done. Unfortunately, nothing was done. And now this is an eDNA study that was done in 2017 by some colleagues. And what they did was this is the historical data that we were just looking at. And they did lines of eDNA samples in ponds going out from this historical area to see where the current distribution of this animal was. And you can see here that the positive eDNA samples have actually managed to reach some ponds right on the periphery of this sampling area. So even this sampling doesn't give us a true idea of the expansion of this population. But it does give us some idea, and it certainly implicates that these animals have reached the Loire River. So this is the Loire River. Those of you who know a bit about France will know that this Loire River actually travels throughout southern France. So there's a direct corridor for these animals now to expand over the whole of France. And we've looked at climate suitability for these animals that suggests that they would, in fact, most of Europe is suitable for their colonization at over 1 million kilometers squared. And in this study, they estimate that the invasive range of this species is now more than 2,000 kilometers squared. So it's now covering a huge area in 2017, whereas back in 2004, it was actually constrained to just a very small area. And so we can see here the rapid expansion of this species, in part because this area is covered in ponds. Okay, so these ponds occur in every field. Fields in this area of France are very small. These ponds are for livestock, for cattle livestock. And so they're artificial ponds, and they absolutely cover this entire area. We also did another molecular study looking at the French population, but not just the French population, but also Portuguese and Italy. So this is, at the time, it was all the European populations that we knew about. And so these three diagrams here represent networks of three different genes. So we can see the three different networks because genes have their own evolutionary history, but they essentially, this gene is not very variable, but it's perhaps the simplest place to start. So the different colors we can see correspond to the different clades that we saw in Southern Africa. And we can see the light gray here represents France, white represents Portugal, and dark grey represents a thin slice here represents Italy. So we can see that here, France, Portugal, and Italy all share this haplotype, what we call a haplotype, this genetic allele. But the French population also have this allele. And when we look at some of the other more variable genes, what we can see is that although Portugal and Italy are generally constrained to one or two of these pie diagrams, the French population is extremely diverse. Even we haven't had examples in Southern Africa of this gene, but we can see there are lots and lots of different examples of diversity of this French population. And in this third gene down here, again, we can see lots of examples of the French population where we haven't even managed to find examples of those genes in South Africa. And so what this is telling us is that that French breeding facility didn't just have one single origin. So they don't have animals that only came from Cape Town or from that breeding facility in Jonkershoek that I talked about at the beginning of the talk, but they actually have examples of animals from all over the region. So they have a huge genetic diversity, and this may go some way to explaining how this particular population has expanded so much and adapted to these Western France conditions that are actually definitely cooler than the Cape and quite difficult for animals if they only came from the Cape, but perhaps with this other genetic diversity that we know comes from that yellow clay, so that other area, those animals actually may have been able to diversify a lot more. So lastly, I'm going to talk about the current research that I'm doing on the gut microbiome. I don't know how much you already know about the gut microbiome, but it's known to be influenced by lots of different things like the weather, the diet, sex of animals and their genetic origins. And then it has influence on their immune systems and lots of different other things, other sites of health, including mental health. So we know the gut microbiome actually has a huge importance for animals and for humans. So it's largely composed of bacteria, fungi, and viruses, and it's involved with these key attributes of animals, including assimilation of nutrition, immunodefense, and behavior. We know that diversity reduces with altitude, and we know there have been a laboratory study of Xenopus laevis, of African clawed frogs, throughout their life history stages. And we know that at the point of metamorphosis here, 83% of the bacteria change into froglets and adults that have very different microbial makeup in their guts compared to young tadpoles. And so we see this life history change, and that's actually relatively important. I've done previous studies of gut microbiome in a toad, and these have shown that that depending on how the animals are transported, whether it be tadpoles or adults, you can have very different results in their gut microbiome. So the preliminary questions for this study were whether there's an altitudinal gradient in the native range, and so we would expect a reduced diversity with elevation, whether we can compare native and laboratory populations, and do lab populations have a depauperate microbiome, and whether the native and invasive populations, are these invasive populations as diverse as the native populations. And so there's some idea of the methodology here. We basically take some fecal matter from the gut of these animals. We use a soil kit to extract the DNA, and then we sequence the DNA. And in the study, we only sequence a single gene from the mitochondria of these bacteria. And so I'll continue after a quick change of venue. You can see here the areas that we sampled in this study. These are invasive populations and one from a laboratory population in the UK, and the altitudinal transect that I showed you before from Laura's study. We used gut microbiome from these animals too. And so the others are Portugal, Lille, Sicily, and France. And the colors here represent the clades, the various clades that these animals were sampled from. And of course, the laboratory population from the University of Portsmouth. So what did we find? First of all, what we found was that the microbiome, or the quantity of bacteria was far more diverse. This is numbers of species or operational taxonomic units found in the native populations compared to samples that we also took from the environment. And then if we look at the laboratory population, you can see there's a big drop in diversity, whereas most of the invasive populations are somewhat similar to the native range, but all slightly lower. If we look at hill diversity, so this is a measurement of diversity, we see exactly the same pattern as we see for actually counting the operational taxonomic units. And beta diversity is a way of looking at how these different populations differ from each other. So these samples here are from the environment. And you can see that even though they're sampled from across the world, they're all clustering very closely together in terms of the numbers of, or the amount of diversity that they have. Whereas this ellipsoid with the points inside represents animals from the native range. So we can see there's a big diversity of animals in the native range in South Africa. However, if we look at laboratory animals, you can see that these mostly eclipse this same area of the native range animals, and maybe even with a bigger diversity in the laboratory. But what we found was that these animals varied. The variation in these animals was actually from room to room across this laboratory. They had different animals housed in different rooms, and these each had a different microbiome. These animals in green here are from France, and you can see the animals in Portugal, even though they have a completely different invasion history, are very nested within that, and so are those from Belgium. And so all of the European populations, except those of Italy, are all clustered, highly clustered together. For some reason, the Italian populations seem to be sticking out as somewhat different. And so when I looked to see the influence of elevation, it came out very strongly, and it included in all of my models to describe the diversity that we got, hill diversity. But the best variables included various bioclimatic variables as well, notably the precipitation of the driest month, so how dry does it get, and the seasonality of that precipitation, so whether it's wet all year round or only wet in summer or only wet in winter. And of course, maximum temperature of the warmest month also played a part. So those were the three significant models which correspond to work that we've previously done on the distribution of this species. I also did this extra analysis. This redundancy analysis tells us how influential these different climatic factors were. And so you can see this, these two seasonal factors pulling in opposite directions, whereas elevation is somewhat less of an important factor coming down. So this is maximum temperature of the warmest month, and the other one is precipitation of the driest month. The last thing was, the last question was to ask which of these taxa actually had their shared, which of the taxa was shared across the different environments. And so we have here laboratory, native, environmental, and invasive. And you can see that most of the environmental taxa are actually also shared by animals. And a big core of the microbiome is also shared with laboratory ones. In fact, the laboratory ones don't have much additional. They just have parts of their microbiome that they presumably brought with them. About half they've managed to retain from the native populations, whereas the invasive populations have acquired a lot more than the laboratory population. So it seems that there is a lot of the microbiome coming in there. Okay, so the last part of my talk is just to talk briefly about what we've been doing in Arizona. A lot of you will be aware, probably better than I am, that the population in the 1970s, I think it was 1979, a certain character from a tertiary institution in Arizona released laboratory animals into several places around Tucson. And this, what was the Arthur Pack Desert Golf Course that I visited first in 1995 and caught animals there, seems to be the only one of these that's where the animals have actually been retained. So I visited as part of my PhD in 1995, and I used this canoe to set traps in two of the water obstacles and caught quite a lot of animals. And this is how it was last year when I visited. This is actually the same shot. You can see the wall here is the same wall here. They've lost the weeping willow and various other trees and have grown some more by the looks of things. But yeah, this is the same water obstacle just last year. And so what are we doing there? Well, the Arizona population has always fascinated me because in summer, obviously the water temperature gets very hot and the animals look very emaciated and unwell when I've sampled there. So one of the first things that I did was to place a temperature logger using the same canoe that I used 30 years before. I rowed out and attached it to a pole in the middle of the lake. And what you can see here is the temperature profile of the water. This first part is at the surface, and then unfortunately the surface one stopped working, so we put this one to the bottom, and that's why there's a slight change here. But you can see that the temperature does indeed get very hot in the summer here, around 37 degrees, and goes just below 10 in the winter temperatures, and then coming back up towards 30 in the summer. And we've got I think another month of data that needs to be collected on this. And this might remind you of something because actually it's very, very similar to the data, the water temperature data that we collected from the coast, from sea level in South Africa. And so those two temperature profiles actually very similar there. So it seems that even though we might think of the Arizona population as being extremely different for these animals, it's almost identical to temperatures that they receive elsewhere in the world. So Casey Perkins, who might be known to a good many of you, or hopefully he's in the audience even now, has been going there facilitating the temperature profile that you've just seen. He's been regularly pulling in that temperature logger. He's also putting out small little audio moths so that we can monitor the calling of these animals and find out when they start breeding. We know from my visit last August that there are large tadpoles to be found during August, but we presume that they actually start breeding much earlier on in the year. And Case is also taking monthly samples of the animals. So what we're interested in there is finding out when during the course of the year these animals are actually feeding and able to store up enough fat so that they can pass through the winter and then start breeding again in January, because during midsummer, certainly in August when I was sampling last year, these animals are looking really emaciated, very, very little fat. And so it seems almost as if every year they're only just managing to survive the summer, and then actually it may be that the winter is their good time. But hopefully we'll be able to find this using these monthly samples collected by Casey. And then I'm just going to quickly wind up by telling you about the past 3-4 years of my research. I've been doing all of these different samples in Southern Africa, but I've visited almost every other population in the world, certainly all that we know of. And this year I was at 2 sites in China, as well as about 4 different sites in Japan. And then prior to that, I was in Chile And of course, I was in California, Washington, and Arizona, as well as these European sites. And it's quite amazing to meet not only the frogs from all these different places, but all the people. So here's a few shots of people. This is us collecting in South Africa. This is Sicily, France, Belgium, and Portugal here. And then here is Chile and the laboratory that I was working with. So these are all Chile at various sites up in Copiapó here. And here is California, Washington, California again, and yeah, that's also California. And lastly, my Arizona trip. And basically, this is part of me saying thank you so much to all these people who have helped collect these animals right around the world. Fantastic to meet so many wonderful people, herpetologists, so many really interesting, fascinating stories. And yeah, it's been a great, great adventure for me to wind up in places like China here and Japan, collecting the same animals from lots and lots of different places with lots and lots of fantastic people. So it only remains for me to say thank you very much for your attention. Thank you very much for listening. If you want to find out more about these studies, then in the fullness of time, everything is going to be published. And I keep a blog on my website, and so you can scan this QR code here, or you can just go to my website, john.measy.com, and I regularly update the blog, and that's where the results will appear first, if you want to do that. Now, hopefully, during the actual course of your meeting, I will be in a minibus traveling from this city called Lijiang further up into the mountains on my business trip. But I'm hoping that I'll have cell reception. So if you do have any questions, I'm hoping that we can get in touch and I should be able to answer them. So thank you very much for your time. Goodbye.